We are now recording. So welcome to those of you that are joining us online at a different date. So today we are talking about chapter 19. Chapter 19 is the chapter about the different senses. So we sort of briefly touched on the eye, and now we're going to talk about some of the more external. We're going to talk more about some external surfaces of the eye. So this is your conjunctiva. Your conjunctiva is going to line the anterior surface of the eye, anterior meaning this way, but it's not going to line the cornea. The cornea is not going to be that external surface. And it's also going to line the inner surface of the eyelid. It is going to be made up of stratified squamous epithelium. So let's break this up. So what does stratified mean? Those of you guys that are here, you have to mute yourself.
Can you hear me?
Yes.
Like multiple layers.
Yes, stratified means multiple layers. Good. And then squamous. So what does squamous mean?
Squamous. Like squished and disc-like.
Yeah, exactly. So flat, squished, disc-like. So this type of cell is made up of stratified squamous epithelium. Epithelia is that tissue that is going to be made up of lots and lots of cells. Not a whole lot of anything else, just basically cells on top of cells. The conjunctiva is also going to be made up of these goblet cells. There's a lot of goblet cells in the conjunctiva. Does anyone remember what goblet cells secrete? What do goblet cells secrete?
Can you hear me? I don't know if I'm right, but is it mucus?
Yeah, it is. It's mucin, and mucin mixed with water creates mucus. So some other components of this external eye were to include the eyelids and also the eyelashes. The eyelashes are not just for looks. They are going to have these important characteristics to be able to help to protect the eyes and also to be able to prevent any foreign materials from being able to enter the eyes. So they kind of act kind of like a broom. If something is coming towards you, they close so they can kind of capture what that is. If you've ever gotten something stuck in your eyelash, then be grateful that that didn't end up in your eye. Some other components, we have this dark circle in the middle. This dark circle in the middle is called your pupil. If we were to remove everything but that pupil would actually be much, much larger. But we have this other muscle that is going to control how much light is going to be able to enter that pupil. That colored muscle that is surrounding that pupil, or that is really on top of that pupil, that's called your iris. That iris, that's that pigmented portion. That's what in some people is blue, other people brown, hazel, green. So the sclera is the white of the eye. This is going to be covered by this conjunctiva. We have this lacrimal caruncle. So remember, lacrimal, what that word means is, it means referring to tears. That's what lacrimal means, tears. And so this is that caruncle where that's where the tears end up going, which we're going to talk about that in the next slide when we talk about how tears work. So this is your lacrimal apparatus. So remember, lacrimal refers to tears. The apparatus is basically how your tears work. We have this lacrimal gland. The lacrimal gland is the gland that actually produces the tears. So this is where those tears are produced. They are then going to be transported to the actual eye right here. Then our eyelid, our upper eyelids, well, both of our eyelids are going to close. And when that happens, those tears are going to be transmitted across the eye. Then that fluid is going to enter that lacrimal puncta right here. That lacrimal puncta just means a dot, basically. And so that dot is where those tears are going to drain out. And then it's going to drain into that lacrimal paniculi right here. And then from there, it's going to drain into this lacrimal sac. This lacrimal sac is eventually going to drain into your nasal cavity through that nasolacrimal duct. So if you've ever been crying and you notice that your nose starts to drain as well, you have a lot of stuff coming out of your nose that's not actually snot, it is actually tears. Those tears come down through your eye and then they get drained through that lacrimal puncture. Whenever you have a tear that actually falls down your face, that's just going to be too much tears for your lacrimal puncture to be able to handle that little tiny hole. The purpose of tears is going to be to primarily lubricate the eye. This is really, really important. And it's also going to help to protect your eye. It's going to help to protect your eye from any potential pathogen. These pathogens could be bacterial infections. And the way that it does this is through this enzyme. This enzyme is called lysosome. And this lysosome is going to be basically an antibiotic. And then just like if we were in an actual class, you guys can feel free to unmute yourself and ask any questions throughout.
Am I muted right now?
No.
Oh, okay. Can you see my video right now?
No.
Okay. I'm good with that.
Okay, so moving on, we've got 3 principal layers that are going to form the wall of the eye. Oh, hi, Kendall. Can't see you or hear you, but I see that you're here. So these 3 principal layers that are going to form the wall of the eye are going to be the fibrous tunic. Here is the fibrous tunic. So what does fibrous mean? What is this word? I'm going to unmute you guys.
Has
fiber?
fiber?
Fibrous mean? Yeah, it has fibers. Yep, that's true. What does that mean as far as what characteristics or what it seems like?
Strong.
Yeah, exactly. So this fibrous tunic is going to be a really, really strong layer. It's going to be this really tough outer layer. I'll mute you guys again. Kendall, you're just joining me. I am muting you guys so that there's not very much interference. But if you have a question or anything like that, you can go ahead and unmute yourself. It should be pretty self-explanatory how you do that. I'm sure it looks different for you guys than it does for me. Okay, so we have this outer fibrous tunic right here. That fibrous tunic is going to be made up of the sclera, which is going to be this part that is on sort of the exterior part of the eye or the white part of the eye. And then we have this cornea. The cornea is going to be this part that is going to be covering your iris and your pupil and then the lens that are behind those. So then we also have a vascular tunic. So the word vascular, so what does the word vascular mean?
Has blood vessels?
Yeah, exactly, it has blood vessels. So having blood vessels is an important part of this. All of these are going to be parts that are going to move. We have the choroid, which is made up of those blood vessels. But then we also have the ciliary body, which the ciliary body is an important muscle that's going to help to control the lens. We have the lens. The lens is going to change shape depending on what you are focusing on. The last layer of the eye structure, this is the most internal layer. This is going to be your retina. And your retina is going to be made up of these 2 different layers. We have the pigmented layer and the neural layer. So let's go into a little bit more detail about these different layers, about these different tunics. That's what the word tunic means, by the way. Tunic means layers. So we have the cornea. The cornea is going to be this top layer that is going to be above, anterior to that iris, that pupil, that area. So your cornea is transparent and it is avascular. So because it's avascular, that means that it does not have any blood vessels. And the cornea is going to receive oxygen and nutrients from the underlying layer. Then we have the sclera. The sclera is going to be the white of the eye. That's going to be on the outer surface of, so it's not going to be where the lens is or where the iris is or where the pupil is. It's going to be where that white section is. So within the vascular tunic, we have the choroid. The choroid is going to be full of those blood vessels, and those blood vessels are going to help to supply nutrients as well as oxygen to the retina. Not only that, but it's also going to help to bring nutrients and oxygen to other components of the vascular tunic as well, such as the iris and the ciliary body. So the ciliary body is going to be composed of ciliary muscles. Those ciliary muscles are those really important internal muscles that are going to help to change the lens shape. Your lens is constantly changing shape, and when they change shape, they're going to focus on something different. So if you're looking far away, if you're looking in the distance, the lens is going to look very different than when you are looking close up at something. So then we also have the iris. The iris is going to be that pigmented part of that anterior eye. And the iris is a muscle. This is that internal muscle that is located within the eye, intrinsic muscle. And this muscle is going to control the amount of light that enters the eye. Really, it's going to control the amount of light that enters the pupil. Can you guys hear me okay? Yeah. And we also have the retina. So the retina is going to be this most internal layer. And we have the pigmented layer that is going to be on the exterior side of the retina. This is going to be attached to the choroid, and this is going to help to it's going to help to receive that energy from the choroid, and it's also going to absorb light energy. So this light energy is going to pass through the retina, and then it's going to help to provide those photoreceptors with vitamin A. Does anybody know where we can get vitamin A from? I thought I unmuted all of you, but I just unmuted all of you. Nope. There we go. It's not letting me unmute. So vitamin A. Does anybody know where we get vitamin A from? Vitamin A comes from orange vegetables and fruit. So these are going to be these like carrots. Carrots are great sources of vitamin A. Also squash and mangoes and that sort of thing. Those are all going to be really, really high amounts of vitamin A. And vitamin A is really, really important for good eye health. So the pigmented layer is going to be that external layer of the retina, and the more internal layer is going to be called the neural layer. So the neural layer is going to be where those photoreceptors actually live. The photoreceptors, so photo refers to light, and receptor refers to some sort of transducer. So this is going to be where the transducer that actually turns the light wavelengths into electrical message that our brain is going to be able to read. Those are photoreceptors. So those are going to be those receptors that are going to receive that light energy, basically. And then also other associated neurons. Those neurons that are going to take, once those photoreceptors transduce that wave energy into electrical messages, then those associated neurons are going to help to move along that message further. So so that our brain is going to be able to interpret it, and so that we are going to be able to say, oh, that looks like an eye. Okay, so let's talk a little bit about some more components here. So here we can see these three layers right here. We've got the sclera right here. The sclera is going to be, again, that white of the eye. We have the choroid. The choroid is going to be that really pigmented section of the or not pig. Well, it is pigmented. It is going to be red and potentially blue. It has a lot of blood vessels throughout it. And then the more internal section is going to be that retina right here. So some other components here. We have this optic disc. This optic disc is going to be this back portion of the eye right here. This is going to be where all of the nerves and the blood vessels are all going to converge. And here is where they will actually leave the eye and make their way to the brain. And after this spot, this is where the optic nerve, the cranial nerve 2, is going to start to form. So this spot right here, this optic disc, is actually going to be a blind spot. If we were going to have lab, we would be able to see that blind spot. You'll be able to see with both eyes. You can clearly see that there is a cross on a piece of paper. But if you cover one eye and you move in a certain direction, that spot actually disappears. Because you are not able to see it, and it just looks like a blank sheet of paper. This is because your brain is going to compensate for that blind spot. Most of the time, it's going to use two eyes to be able to compensate for that blind spot, but when you have one eye covered, that blind spot is going to just appear blank. But it doesn't look like it's a space that doesn't have anything in it. Your brain is going to use what's surrounding it to make a guess as to what's actually there, which is why when that cross disappears, it just looks like the rest of the paper. Really, really interesting, and it's unfortunate that we aren't able to do that. You can read through your lab book at home, though, and you can try to do that on your own if you have the supplies needed. Okay, so for the ciliary muscle, the other parts of the eye that we're going to talk about, we have these ciliary muscles right here. So you can see the ciliary muscle here. You can see how it has these sort of string-like things that are going to attach to the lens. This is the lens right here. So the ciliary muscle is going to maybe pull really tight, and when it pulls really tight, then it's going to pull those strings, and it's going to make that lens sort of flatter. Likewise, if it relaxed, then that lens would be then more concave. This outside, very protective covering is your cornea right here. If you've ever heard of somebody, maybe they had really severe eye pain, and they had to go to the eye doctor because they got their cornea scratched, something like that. That's a fairly common thing, and that's because your eyes are going to be very sensitive, and that's the cornea's job. The cornea's job is to be able to be scratched and whatnot, so that it doesn't actually scratch the iris. You don't want to scratch the muscle behind the cornea. Instead, we want to scratch this. Ideally, we would scratch nothing, but if there's going to be an injury, it's a good thing that it happens to the cornea. So then the iris is going to be that pigmented section here that's going to be made up of those intrinsic muscles. Those intrinsic muscles are going to include the sphincter pupillae and the dilator pupillae. Sphincter pupillae is going to act kind of like a sphincter, and it's going to allow for it's going to close actually that iris to make less light come in, and then the dilator pupillae is going to dilate that pupil. It's going to cause that pupil to become larger. Let's take a look at that. Here's the sphincter pupillae. When that sphincter pupillae is contracted, then that iris is going to become much larger. That pupil is going to become much smaller, and this happens when you have really bright light. This is going to be part of parasympathetic innervation. So, unmute you guys. What does parasympathetic innervation mean? Parasympathetic. I heard something. Oh, try again. Parasympathetic. So the parasympathetic nervous system is, can you guys hear that echo? Just muted you guys. Yeah, okay. So the parasympathetic nervous system is going to be the nervous system that is also called the rest and digest nervous system. So this is going to be your normal nervous system that's in play most of the time. The sympathetic nervous system, this is going to be that fight or flight nervous system. Those are the two divisions of the autonomic nervous system. So when an individual is having this fight or flight response and they are maybe having, there is a bear that's going to be all of a sudden appear in their cave. We're still talking about evolutionary times here. And when that happens, then the pupils are going to dilate, which is also why it's very common for individuals to have dilated pupils if they're on certain types of drugs as well. Because many types of drugs, especially ones that would be considered uppers, they are going to work with the sympathetic nervous system to cause that sort of fight or flight response. Okay, let's talk a little bit more about our lens. So the lens, here is the lens right here. This is going to be a transparent and deformable structure. It has to be able to be deformable so that it can focus on different types of objects, objects that are different distances away. This is going to be suspended behind the pupil, and this is going to be suspended by these things called suspensory ligaments. So these suspensory ligaments are going to be these string-like features right here that are going to be between the ciliary muscle and the lens. These suspensory ligaments are going to be attached to the outer capsule of the lens. And as the tension is going to change based on how the ciliary muscles contract, that's going to change the shape of the lens. So when you are looking at something that is far in the distance, perhaps some mountains that are quite a distance away, our lens are going to be flat. So those ciliary muscles are going to be relaxed, and that's going to allow for those lenses to be flattened. If you're looking at something close up, maybe at your watch or something like that, then those ciliary muscles are going to contract. And as they contract, they're going to become closer to the lens. So as they contract, they kind of get bigger. That's how muscles work. And when they become bigger, they're going to move closer to the lens, which is going to cause those suspensory ligaments to be relaxed. And as those suspensory ligaments are relaxed, then the lens is going to go to its sort of normal shape, which is going to be a very spherical shape, kind of like a dome shape. So when it's really far away, when you're looking at something far away, those muscles are going to relax. When they relax, they're going to kind of spread farther away from those lens, and therefore those suspensory ligaments are going to be very taut. When that happens, it's going to cause that lens to go from this disc shape to a more flattened shape. As those as those ciliary muscles become more taut, more contracted, then they're going to move closer, and then that dome shape is going to be more likely, or going to be clearer. Does that make sense? It's kind of confusing because it's easy to think about the suspensor or the ciliary muscles being relaxed, meaning that they are closer to the lens. Okay, so the ear is going to contain these, so that is the eye. So we have talked about the eye now, and now we are moving on to the ear. The ear is a really cool organ because it not only has to do with hearing, but it also has a lot to do with equilibrium. Equilibrium, what that means is it kind of means balance. So the ear is going to contain both of those organs that are associated with equilibrium and also. The ear is divided into three distinct anatomical regions. We have the outer ear. The outer ear, also known as the external ear. This is going to be the type of ear. This is going to be the part of the ear that you're able to see if you look in with one of those scopes that the doctors use. That is what they're looking at primarily. If you use a Q-tip to clean out your ear, you're only going to be cleaning out in that external ear. Then after we pass, this is the eardrum right here. The eardrum is also called the tympanic membrane. So after we pass the tympanic membrane, that is when this middle ear is going to start. The middle ear is going to be a very small section that is also associated with hearing, as is the external ear. All of these features, your auricle, your external ear here, as well as this external acoustic meatus, these are all going to help to funnel those sound waves and help to make it so that you are able to hear as well as possible. And then those sound waves are going to enter this eardrum and they're going to pass through these three bones that are called the auditory ossicles. Those auditory ossicles are going to be the main components of that middle ear. Then we have the inner ear. The inner ear has part of it that is associated with sound. So as that sound moves through those auditory ossicles, those really, really small bones in your ear, it's going to move through this cochlea. This cochlea is this seashell-shaped organ right here. And that is going to be where those sound waves are actually going to be transduced. That word transduced, that just means moving from some form of energy into another. In this case, the sound waves are going to be transduced from sound waves to electrical message that our brain can read. Another part of this inner ear is called this vestibule right here. This vestibule has these three semicircular canals right here, and those are going to be that's going to be the organ. The vestibule is the organ that's associated with balance and equilibrium. I'm going to talk a lot more about this in a minute. All right, so let's first talk about our external ear. We have this auricle right here. This auricle is made up of elastic cartilage, and it is funnel shaped. This funnel shape is going to help it to be able to, it's going to help to funnel those sound waves into the external acoustic media. It's going to end at this tympanic membrane. Again, tympanic membrane, that is the anatomical word for your eardrum. A timpani, if you've ever heard of that, that is a type of drum. So it basically means an eardrum. So deep within this external acoustic meatus, right here, we have these glands that are going to produce this wax-like secretion called the cerebrum. Cerebrum, that is going to be known as your earwax. All right, so moving on. We also have this thing called a middle ear. So the middle ear is going to be more internal than the outer ear. And this is going to start at this tympanic membrane, at this eardrum right here. And then it's going to move from the tympanic membrane through these auditory ossicles. So the tympanic membrane is going to have, it's going to be the start of this tympanic, excuse me, it's going to be the start of this tympanic cavity right here. So this tympanic cavity is going to be filled with this substance that is going to be a fluid-like substance that's going to help with the movement of those sound waves. This is also going to connect to the nasal pharynx. So if an individual is having a lot of issues with their sinuses or with their nasal pharynx is going to be that nose throat region. And when if an individual is having issues with if an individual is having an ear infection, a middle ear infection specifically, they are often going to have issues with drainage of their nose. That's because it does have this tube that connects there. So we have these three auditory ossicles. We have the malleus. The malleus is this feature right here that looks kind of like a hammer. Here's the handle. Here's the hammer part. Here's the anvil right here. The anvil is called the incus. That's another word for anvil. Right here. And then we have the stirrup, which is the stapes right here. Here's that stapes. It looks just like a stirrup on a horse. So those three, the malleus, the incus, and the stapes, those are the smallest bones in the body, and they are going to make up those auditory ossicles. The stapes is actually the smallest bone in the body. All right, so moving on to the inner ear. The inner ear is located in spaces of this petrous part of the temporal bone. So petrous, what the word petrous means is it means a slit, a lot of holes throughout. So here we can see there are many, many holes, kind of like little tiny sinuses that are going to be throughout this temporal bone. So these spaces that are going to make up this inner ear, these are called the bony labyrinth. So here we have this bony labyrinth right here. The bony labyrinth is also called the semicircular canals. Within the bony labyrinth are tube-filled spaces, the fluid-filled tubes rather, and these spaces that are called the membranous labyrinth. So the membranous labyrinth are going to be the space within the bony labyrinth, within those semicircular canals that are going to be filled with fluid. So this is, we're talking about your vestibule right here. This is your vestibule, and the vestibule is going to be the organ that is associated with, it's going to be associated with balance and equilibrium. So this fluid is all going to react with water, or react with gravity rather. And as it reacts with gravity, that is going to, it's going to be able to allow you to be able to know if you are leaning sideways, and it will be able to sense those differences between those ears especially. So we have the receptors for that equilibrium. Those are going to be housed within the sensory epithelium that's going to be lining that membranous labyrinth. We have a lot of these different hair cells that are going to act as these receptors, and they are going to be housed within this membranous labyrinth. Remember, the membranous labyrinth is going to be this pink feature right here that's going to be in the middle of the bony labyrinth. The whole thing is really the bony labyrinth. And then internal to that bony labyrinth, that is going to be that membranous labyrinth. So the bony labyrinth is going to be partitioned or separated into these three distinct regions. We have the vestibule. The vestibule is going to be this part down here of the bony labyrinth, right here. And the vestibule can be broken up into these two different regions. We have this utricle right here and a saccule over here. We also have the semicircular canals up here. And then we have the cochlea. The cochlea is going to be the organ of hearing. The vestibule and the semicircular canals, those are going to together create this area that's called the vestibular complex. The vestibular complex is going to be this whole organ that is associated with equilibrium. Whole organ, right? That whole organ is the vestibule. Right. Let's talk a little bit about these receptor cells that are located within those semicircular canals. So we're not going to go into as much detail as we would have originally, so don't worry about these lines here. I kept them in this slide and I just crossed them out so that you know that they are not going to be tested out. I'm not going to put the difference between stereocilia and kinocilium. But just know that these hair cells are going to be located within the semicircular canals, and those hair cells are what are going to sort of tell what is going to be affected by balance. And how they work is they are going to work with these otoliths. And these otoliths are going to be these stone-like structures that are going to be within this gelatin. And so what happens is that when you tilt your head one way, any direction, all of the gelatinous material are going to move in that direction. They're all going to follow gravity. That's sort of, they all are going to work closely with gravity. And when that happens, they are going to bend those hair cells. And when that happens, those hair cells are going to recognize that what's happening, and then they're going to be able to, those especially those otoliths, those small stones, they are going to either they're either going to move those hair cells, which will cause those hair cells to then transduce that message, basically. And transduction is going to be the movement or the transformation of one form of energy to another. In this case, it's going to be the transformation of physical energy, mechanical energy, the movement of those otoliths to this electrical message that are bringing. So I should have crossed this one out too, this line right here. Again, we're not going to go into the detail, into the difference between the stereocilia and the kinocilium. We are going to have to cut a fair bit of material from our lectures overall to account for this different type of lecture. Okay, kind of went over all that. Okay, so let's talk a little bit about rotational movement. Rotational movement is going to be the movement that happens when you spin. And when that happens, an individual usually gets dizzy. They get dizzy because these hair cells are able to adapt really, really, really quickly. So if you start spinning, and then those hair cells are going to learn that this is basically the new normal. And then when you stop spinning, the hair cells have to readjust to this new normal. It's the old normal. Okay, so let's talk a little bit about this rotational movement. So we have these semicircular canals here. The semicircular canals are going to move, or they're going to come off from the utricle right here. And then we have the receptors within those semicircular ducts. They are the ones that are going to control the rotational movements of the head, or they're going to detect and they're going to transduce those movements. So when the head moves, we have this this gelatinous type of stuff right here called the cupula, and the cupula is going to then move in one direction or the other. So when your head is rotating, that cupula is going to be moved to one end. And when that cupula moves, it's going to basically cause those hair cells to sense that movement. And that's what's going to allow us to have that sensation of spinning. This gelatinous material that's on the outside of the cupula, this is called endolymph. And there are many hair cells that are going to be associated with these different nerves, these neurons rather, and then together those neurons are going to create this nerve. Okay, so that is equilibrium in a nutshell. So let's talk about some structures for hearing. So the cochlea, this is that seashell-shaped organ right here. If you can see my face in this video. Okay. So this seashell-shaped organ, this is called the cochlea, and this is the organ of hearing. This is where those sound waves are going to be transduced to electrical energy that our brain can read. So this membranous labyrinth is going to run through the cochlear duct, and as it runs through the cochlear duct, it's going to basically it's going to run its course along through this duct. So let's say we have this seashell right here, and let's pretend it's kind of like a croissant that you have wrapped up in a circle. So then you can unroll that croissant, right? So pretend that you're unrolling this cochlea, and this is what we're looking at right here. This is that cochlea that's unrolled. So throughout this cochlea is going to be this membranous duct, and this membranous duct is going to be split into these two different chambers. We have a scala vestibuli on the outer portion right here, and then going around through, then we have the scala tympani over here. Let's talk a little bit about how these sound waves are going to be transduced. So these sound waves are going to be funneled into the ear through your auricle right here, and then in through that external acoustic meatus. Then the tympanic membrane, the eardrum, is going to sense those vibrations, and those vibrations are going to be then sent through those auditory ossicles, where they'll be a little bit magnified, amplified. The stapes, this is that stirrup structure that is going to be the first step to the cochlea. That's going to be the window between the middle ear and the inner ear. And so the stapes is going to send those messages, which are in sound waves format right now, through the scala vestibuli. So then those sound waves are going to move through the scala vestibuli, and they're going to continue for as long as they can, depending on how much energy they have. So depending on how much energy they have, they will be transmitted through somewhere at some point. And then that is how we are able to detect different pitches of different sounds, different levels of amplification. All right. We also have different levels of frequency. And so we have different levels of frequency, and this is going to determine basically how far those sound waves are going to be able to travel through the tip. So the louder sound waves are going to be able to travel through here, to get to this round window. Depending on, that's the goal. That's if it's loud enough. But where they're going to cross right here, this is going to depend on their pitch. So the really, really high-pitched sounds, they are going to have a lot of energy associated with them. And so therefore, they're going to end right here. If an individual has really, really low frequency, this is a really deep voice, this is going to move further along, and perhaps it's going to end further within that cochlea. Remember, this is that cochlea. This is going to be that cochlea that's unrolled. And again, it's going to be that seashell shape. So the lower frequency sound waves, those are going to have that low pitch, and those are going to terminate closer to the tip of that cochlea. So the tip of that cochlea is going to be closer to this middle part. Farther along goes, the lower the pitch. Does that make sense? Kind of a lot with frequency and amplification of sound or loudness. Okay, so cochlear implants. These implants are going to enable a lot of deaf individuals to be able to hear. And so basically what happens is we have this transmitter that is going to be in your skull, basically. And then we have a receiver and an antenna that's going to be located within your skin. So the transmitter is going to be something that's clearly visible if an individual removes their hair. Then this receiver and this antenna are going to be then attached to your cochlea through this transmitter lead. And this cochlea then is going to, or this transmitter lead is then going to basically act as the cochlea. It's basically going to be a cochlear transplant. Okay, so I'm the only part of this development of the eye that I'm going to go through is that this happens during the 4th week of development is when those original eye ossicles are going to form. And then eventually, and then by about week 20, that is when the actual tunics of the eye are going to form, and that's when it's going to start looking like a real eye. Before week 20, it's going to be very, very, very immature. But the eyes, the actual formation of the eyes are something that develop really, really late. And so individuals that are very premature often have a lot of issues with being able to see properly. And then the ear development happens much earlier on. So this is going to start in about week 4. So remember week 4, that's going to be the first time that an individual is going to be able to pick up a positive pregnancy test. And then we are going to, moving on those next couple weeks, this is all during the embryonic phase. The development is going to, the actual cochlea is going to develop during this week 8. This is that C-shelled organ that is going to be fully developed by about week 9. So by about week 12, that is going to be when the baby is going to be able to actually hear, pick up sounds. That isn't to say that that baby will be able to interpret those sounds, but it will be able to at least